Our guests today talk about the next big transition in modern medicine, the move to human proteomics. Start your tricorders. It's Futures in Biotech.
Netcasts you love.
From people you trust.
This is Twit.
Bandwidth for Futures in Biotech is provided by Cachefly at cachefly.com.
This is Futures in Biotech, episode 75. It's time to start the human proteome. This episode of Futures in Biotech is brought to you by Kaleidograph, a thoughtfully designed graphic and data analysis application for research scientists as well as those in business and engineering fields. Try Kaleidograph for 45 days, risk-free, and receive a 20% discount if you purchase Kaleidograph within the next 60 days. Visit www.synergy.com/podcast.htm for more details.
I believe that biotech is the next frontier.
Probably the greatest intellectual revolution that's ever taken place in man's history. DNA is the code for life. We're actually beginning to understand how life works, which I think is something that's mind-blowing in and of itself.
There was going to be a genetic component to aging.
How long was there the extension?
About 30-40% for humans. That would equate to something like 20 to 30 years.
How close are we to actually having a therapy?
Ballpark, 10 years.
It's potentially one of the things that'll end up rocking the world the same way that people said, oh, the sun's the center of the universe, oh, this and that and everything. And now here's somebody who can come out and say, hey, look, here's how we compare it to our closest evolutionary relative.
Welcome to Futures in Biotech. Today, we're going to talk about proteomics. This is going to be one of the hardcore versions of the Futures in Biotech. We're talking to two really smart guys. They proposed a paper, and this is what got me to let's start over on 3, 2, 1, if we can. Welcome to Futures in Biotech. I'm Marc Pelletier. Today we have a very interesting show coming on, and we're going to talk about proteomics. And I believe this is the next stage of modern medicine. And our guests proposed a paper. It was in September in Nature Methods, and they suggested that mass spectrometry was ready for the big time. This is kind of a historical paper, in my opinion. It's much like the early work that led to initiating the Human Genome Project, except in many ways it's more ambitious. It is really hardcore. So put on your thinking caps and hang on tight. Just give you a heads up too, it took me about 3 years before I started understanding what one of our guests was talking about. He never dumbed anything down. And it was a great sort of mentoring strategy in that it allowed us to try to get to his level and understanding of the human anatomy and how we work. So our first guest is Dr. John Bergeron. He's professor and chair of the Department of Anatomy and Cell Biology at McGill University. He's a member and an active member of the Canadian Proteomics Network. He's a former president of the Human Proteome Organization. And co-founder of Caprion Proteomics. He invented their main technology called CellCarta. He was also a guest on Futures in Biotech episode 12, and I think it was 17, the Singularity episode. Welcome to the show, John.
Oh, I'm glad to be present again, Mark.
Our next guest also is at McGill. His name is Dr. Tommy Nilsson. He's professor in the Department of Medicine. And he's also a Canada Research Chair in Proteomics and Systems Medicine. Welcome to the show.
Thank you very much. I'm very happy to be part of this.
Well, this is, it took some ambition and guts and foresight and sort of a very positive outlook to propose what you guys were proposing. So perhaps you could tell us a little bit about how you got involved in proteomics and where you see it going and what led to writing this paper. John?
So, okay.
Tommy? It's okay. Tommy.
I think John is the person with the more history on this. We have, can just mention, I've been pulled into this during the last 5 years, much through John Bergeron, and we have had a series of workshops in Barbados. Which sounds very exotic, but we actually don't see much of the sun when we are there and we're working. This is in January. The thing which we have been doing is to try to come up with a project to move proteomics forward. And this was a call made by the European Commission. To try to get the proteomics community to implement standards and show that they can work in a coherent manner. And that was a starting point. And from that, when we met and we discussed, etc., it then evolved into something that became a prelude for a Human Proteome project. And that has had a set of workshops in Barbados and other meetings, people meetings, etc., and really to try to define what is a Human Proteome project. It's not really obvious to everyone what can be done in a Human Proteome project, etc., and how we should go about. And we are not, how to say, at an end point in terms of defining this. We are still in a very early stage, and I like always to point out the history of the Human Genome Project, which started sometime in 1985, followed by a very, very important workshop in Los Alamos in 1986, which produced a white paper on the human genome. And you can see on the white paper that we have produced, which is on the HUPO webpage, that we are in more or less at the same stage and in the same spirit. So I think we will get to the details of what we are proposing, but I'd like to everyone to have that in the back of their mind that we are at the discussion and design stage at the moment. John?
Yeah, go ahead, John.
So you asked about how we got into this and why we're doing this. So Tommy and I are both trained as cell biologists, and we've had the great opportunity to figure out some machinery in the cell which no one else had stumbled across. And we had done this before. We're very lucky to do this at relatively early stages in our career. And we focus on the inner workings of the cell by looking at the different compartments of the cell. And it didn't take us long to figure out that mass spectrometry seemed to have been designed specifically to answer all our unknown questions about what made up the proteins in these structures and how the cell was put together and how the cell worked. So that's the strategy that Tommy and I have followed. And by doing that, we've gained tremendous biological insight through the proteins that we've been able to characterize. But on the other hand, we've stumbled across what we think is the strategy that can be used to map the entire human proteome and give functional significance to the proteins as we're characterizing them. So technically, the issue is one of reproducibility, sensitivity of the protein characterization in the samples that you're giving to the mass spectrometer. And the mass spectrometer has such a fabulous mass accuracy that it will actually read out for you, it does it in an indirect way, the primary sequences of peptides in proteins, because that's what you present to the mass spectrometer, are the peptides, in a quantitative way, and it will do it over and over and over again for you, 24 hours a day, 7 days a week. So this is fabulous, providing you can give it the kind of samples that match the characteristics of the mass spectrometer. And those characteristics, the greatest limitation, is a limited dynamic range of about 3 orders of magnitude.
Let me ask you, before we get into the details of sort of the technology, I'd like to say take it down to, or maybe up to 37,000 feet, and then we'll bring it down slowly a little bit. And the reason why I still want to know the context of how this compares to the Human Genome Project. Has the Human Genome Project made it easy for you or hard for you? And then how does this compare to perhaps, gene expression profiling, which is the gene chips, and then you've got proteomics? Maybe start with the first question. How does this compare to the Human Genome Project? What kind of information versus the Genome Project will this provide?
So jump in, Tommy, whenever you want. So one of the major deliverables of the Human Genome Project is an estimate of how many genes we have in each of the cells in our body. And that estimate has actually only been nailed down as of late 2008, 2009, to be roughly 20,300 protein coding genes, and this is still undergoing change and reassessment. So
we have
that's
23,000
20,300. 20,300.
So we've got 23,000 different
20,300.
20300.
Yes.
23,000.
So 20,000. Let's just round it up to 20,000.
By the way, I'm going to mention it to the audience right now because you guys are in Montreal. We have two people in Montreal. We're in I'm in Cleveland, and we're piping through Skype in California. So there's about a 1 second lag. So we're kind of trying to interject. It sounds like a major interruption. So I apologize for interrupting with this lag. But so we have 20,300 or 20,300.
Yeah, let's just round it up to 20,000.
All right, roughly 20,000 parts.
That's right. And so that is a major deliverable of the human genome. However, the evidence...
John, so do you want to put that in context with the fruit fly?
Yeah, go ahead.
No, I'm just we don't have wings and we don't have 6 legs and 1,000 eye facets, but I think we put it in context of 20,300 genes. The fruit fly has something which you could average up to 15,000. So that, I think, gives you a sense of what the genome has done for us, because it really has defined a number which is not, I would say, astronomical in terms of having a genome, a human genome, as a reference. But you have, at the same time, to appreciate the complexity that comes within all the different variants of each gene, which we will come to later. But that sets us apart from the fruit fly and other organisms.
So we're 5,000 genes more complex than a fruit fly.
Yes.
25% more complex.
But let's consider what we don't know. So we have an estimate of 20,000 genes in the human, and that is a deliverable from the Human Genome Project. The question now is, what evidence do we have for what these genes are doing? And let's take the smallest chromosome, chromosome 21, which is one of my favorite chromosomes, and look at the databases, including this most pristine database, which has given us our best estimate of how many genes are in the human genome. And there it predicts to have slightly more than 200 genes in chromosome 21. That's all. However, there's only evidence for 148 of these genes that actually make protein. This is not to say that the other genes do not make protein. It's just that there's no methods to detect them, even including, for many of those genes, RNA expression. So you have to appreciate that in the 250 or so different cell lineages in our human body, many proteins will be long-lived and will be present in a small number of cells, and they will, by definition, be very difficult to capture if you're doing RNA expression profiling from whole organs or tissue or fluids or whatnot. However, if you're able to reach into those cells that are expressing the protein and grab the compartments where they're present at highest concentration, which is what Tommy and I are paid to do, then you get them at very high abundance. So you can now go in and take these 20,000 or so predicted protein coding genes, of which approximately half have some evidence for a protein, and now actually figure out where each and every protein is expressed and directly where it's located, what its protein partners are, and most importantly, what its splice isoforms are, its state of glycosylation, and its state of phosphorylation. These are the things that we can do right now with proteomics. Wow, and that is quite different to what the human genome has given us, which is a fabulous foundation.
So it looks like you picked the right horse to follow, right, over genomics.
I mean, but that's just what we do as cell biologists, right? We follow the technology which gives us the most information with the samples that we have.
I guess if you're looking at the genome, you're always questioning, oh, well, here's a new gene. It's really interesting, but what does it do? I don't know. But if you take it with your approach, take the cell, crack it open into its multiple parts, then crack it open until its atomic molecules or to the molecular level, you know what it is by the fact that where you got it, right? If it's in the kidney and you cracked open the kidney cell, cracked open the kidney organelles inside the cell and then separated the proteins, you know that they're all kidney.
Yes, not only that, but also by doing that methodology, you now have the proteins at a very high abundance, fitting the technology. Tommy actually put in this Nature Methods paper, had a very nice analogy to how you would follow this strategy using the analogy of building a skyscraper. So I'm going to let Tommy take you through the skyscraper analogy, if that's all right.
I will do that. I just want to make one small point, and that is that, I mean, you say we're betting on the right horse, etc. The thing is, first of all, as John also pointed out, the genome is really the foundation, and without the genome, we would have no reference. And we would have been talking, well, maybe there are 50,000 different ones or 100,000 different ones as they thought before. Really, it's because of the genome that we can place it now around 20,000. But the genome and genomics plays a very, very important role in defining all the different types of variants that exist for each and every gene. These could be, I'm not now thinking about the isoforms, etc., but I'm thinking about in every person we will have small mutations that sets each gene apart from individual to individual. These are called SNPs. And of course, knowing these SNPs and having them mapped out gives also then, in many cases, a relevance at the protein level. And these are very much the foundation for disease and disease biology. So having the genome, having the genomics, having the transcriptomics, having the proteomics, and there is also metabolomics. You really have to see these as complementary approaches, and I don't want to say that one of these is much better than the other. They really are there to complement each other. But with proteomics, we can do a lot that you can't do with the other techniques. That's a given. And in disease relevance, in drug development, etc., most, if not all, of the treatments that we have, the drugs, etc., they are against proteins. So proteins are, in that respect, very relevant in terms of the clinical and medical and biomedical aspects. So back then to how we envisage how you could go forward in trying to set up human proteome, we really then fall back to the genome, and that would be like, as John said, and what we proposed is like a skyscraper, and that is the building itself, is the genome. And what every room in that building represents is one of these 20,300 genes. So if you have that building in place, then over time, if it's within the next 5 years or 10 years or 100 years or 1,000 years, depending on how much details you want to put in, you can furnish each one of these rooms in any way you want. And that is the whole rationale for what we call taking a gene-centric approach to the Human Genome Project. So that is, I have a engineering training as a background, and that's how I think about things. And I think this, in this way, we can make sense of the enormous complexity that a human proteome really represents.
One thing that I'm thinking about, if I was in the future and I was coming back in time, where would I want to stop and feel safe in terms of modern medicine? If I went back into the 90s where we had, or late 90s and early 2000s where we had the human genome, I'd be scared if I'd come from an era that had the entire proteome. Would I be fair to say that the true transition in medicine will happen once we get every element of detail from the Human Proteome Project? Is this where it's going inevitably?
So go ahead, Tommy.
Yeah, I was waiting for you, but okay, I'll just give my take on the whole thing. If you come back for the future, it's really the modern medicine, as we call it today, which will be not be modern 20 years from now, because a lot of things will have changed. And the one major thing that will change is that we're going from prescribing drugs that are general, that will fit, one shoe fits all, to having drugs that are more tailored to the individual, because everyone is not the same. So everyone will not respond in the same way to these more general drugs. So if you want to increase your efficacy, you want to increase your effectiveness overall in treatment, even increase your abilities to diagnose and for prognosis, then we are moving into something which is called personalized medicine. And we really there need to very accurately phenotype the individual patient. And there, of course, you would ideally like to use all that genomics gives you as a possibility. So if I came back from the future, I would then be able to tell that 10 years from now, or 5 years from now, or 20 years from now, we can do this. We can have individuals' genome on a chip. We can do very nicely the transcriptome for particular tissue of interest, and we can do the blood, and we can do the proteome of the tissue, etc., in such a way that we can, in the best possible way for that particular patient, then define the type of treatment that the patient should have.
So maybe I can just bring us down to something really defined. If there's someone coming back from the future and you want to chart progress in human medicine through these technologies. So I want to keep taking us back to chromosome 21. Chromosome 21, again, is the smallest chromosome. It has less than 200 proteins. We can figure this out right now. But let's look at the challenges. Everyone, I believe, in your audience is aware of someone, or is certainly aware of trisomy of chromosome 21 that leads to Down syndrome. And even though there are screening procedures in place, we still have about 1 to 2 births per 1,000 in North America, which is still enormous. And immediately we see that these infants have cardiovascular defects, but they also, a separate cohort of them, have leukemias and a special subtype of leukemias, and they have problems with their immune system as well, especially with respect to the thymus. And yet we have no mechanistic explanation of why these diseases are progressing due to chromosome 21. Remember, it's the smallest number of genes, right?
Right. And can you explain what trisomy is exactly? What happens to that chromosome?
Trisomy would be 3 copies of the chromosome instead of the normal 2n. You would have 3n of the chromosome, and so now it's simply having more proteins expressed. That's all that happens when you have trisomy of chromosome 21, and so consequently, for only that chromosome, remember it's only 100 and right now about 200. It's only 10% of the human genome. And yet we have these devastating effects on these infants for which we have no mechanistic explanation. But by understanding the proteome of what those genes are encoding for, and remember, we still have at least 50 to about almost 50% of the genes that we have yet to uncover in terms of what the proteins are doing for that chromosome, we should be able to figure out mechanistically what's going on with these leukemias, these acute myelogenous leukemias that these patients have as a consequence of this, and even get into the broader population of patients that have acute myelogenous leukemias that they get not from Down syndrome, but just normally as they develop cancers, because what's less well known is a large proportion of those patients have only in their cancers trisomy or tetrasomy of chromosome 21, not in the rest of their cells. So there's a link between chromosome 21 and these chronic myelogenous leukemias, where we can now elucidate exactly how that leukemia is put together and what's going on with it to give us some insight into cancer. The other aspect of this is that these patients, the Down syndrome children that have the leukemias, never develop solid tumors. So lurking in that chromosome is some sort of a powerful suppressor of solid tumors. So these are unbelievable benefits that the proteome can deliver us right now, even if we just launch the mapping of the proteins from chromosome 21, which, as I said, are only a couple of hundred or so to be uncovered. So certainly you'll be able to chart, if the chromosome 21 project is completed, exactly what mechanistic insight and clinical relevance have we obtained from mapping the proteins in that chromosome in this gene-centric effort, in the effort to fill up the rooms in Tommy's skyscraper, where now 200 or so will be filled up, with immediate clinical relevance developed. And that would be the paradigm for the impact of this kind of information on the clinical and biological community. Because so far these problems are unresolved, both at the basic biology level and at the clinical level.
You have to get started yesterday. I'm glad you guys start going through this.
We're ready to go.
Okay, so let's take a minute. We're going to take a minute to thank our sponsor, and then we'll come back and talk about the chromosome 21 version of the pilot project of the proteome project that you want to present. I think it's an amazing first step. But before we do, I'm going to thank Kaleidograph for sponsoring this episode of Futures in Biotech. Kaleidograph is a thoughtfully designed graphing and data analysis application for research scientists, business professionals, and engineers. It produces publication-quality graphs and easily converts the most complex data into a functional display. Statistics, linear, non-linear curve fitting, and the ability to produce precise graphic visualization of data all make Kaleidograph powerful and flexible. Since 1988, Kaleidograph has been an easy-to-learn graphing and analysis program with a surprisingly affordable price. I've actually been using it probably since 1996, and it was so easy to use and actually so powerful that I never actually got around to learning Excel to any great extent. Sure, I could flip some cells around, but if you need to do mathematics, you need to present scientific data, you need to present convincing data, you really need a solid application. This was not expensive. Every graduate student should have it. And another thing that it does really, really well is curve fitting. If you ever try to do curve fitting in Excel, it's horrible. You're just going to bang your head on the wall. You're going to spend 6 or 7 hours looking for a plugin on the internet through Google, and you'll always get to, you'll get a polynomial, but what if your model isn't a polynomial? It's only a handful of tools. Well, Kaleidograph has them all. You can model with the equation you want, easy code. They give you a whole bunch of equations to start with. So if you want to do curve fitting, it's absolutely fantastic. We use it in the lab every day. We use it as our primary tool. It's in our workflow to analyze data from cell-based assays. So it's really, really fun. You can do Michaelis-Menten stuff. You can do anything with it. So try Kaleidograph for 45 days, risk-free. And if you purchase Kaleidograph within the next 60 days, you'll receive a 20% discount. Visit www.synergy.com/podcast.htm for more details. All right, that's done. So we're good. We're good to go. So, John, that was very nice explanation as to some of the power or some of the information that could be acquired from doing the human proteome. Am I right to say that the human proteome would be kind of like the Google map of the human body down to the atomic level?
Tommy, I'll let you handle that with respect to your skyscraper model.
Okay, I want to point out that when we talk about proteomics, we have different approaches within proteomics, and both John and I are very much in favor of using mass spectrometry for proteomics, and that the reason for that is that we get immediate insight into the details of each protein that we are analyzing in our sample. But complementary to that, within the realm of proteomics, there are other technologies or approaches, and one very big approach right now is the one undertaken in Sweden through Matthias Julen, called the Human Atlas Project. And there they've taken upon themselves to map every protein in the human body, in the different tissues, using pristine antibodies. And this is a very ambitious undertaking. And what we, in our discussions in Barbados, when we drafted this white paper, we envisaged the Human Proteome project going forward with a couple of different engines. The mass spectrometry will be one engine, antibodies will be a second engine. We also have efforts where we look at protein-protein interactions, which will be a third engine. And somehow we will then not just distill the relevant information, but try to synthesize the information in a complementary way through bioinformatics. And that then becomes a knowledge resource then, which we would then term the Human Proteome Project, but constructed in such a way that we can add more and more information as we move forward. So I think it's important to keep that in perspective, because a human proteome project will only be successful through the applications that will come from this. And we need to know where proteins are within the cell, within the tissue, within the organs, within the body. And there, antibodies plays a very important role. But at the same time, we need to know exactly which protein we are looking at, what type of modifications are on the protein, etc., in that particular place. So the two are very synergistic. Now, how do we then envisage this moving forward? We had, and as I said in the beginning, we had a call from the European Commission to put together a community effort. This is the spirit today, and I think will continue, that we will pool resources across the globe and then try to do our bits and pieces. The Chromosome 21 project in that context is a pilot project. It's a proof of principle that you can deliver immediate value, that we can show that we can work together, we can show that we can have synergy from the different type of proteomics approaches as we have. So at the same time as we are speaking now, Matthias Julin, through his antibodies efforts, are mapping the proteins that are in chromosome 21 at the antibody level. So the two together, then we will have the spatial organization will have a very good idea of where the proteins are expressed. And then we have the mass spec approach, where we go in and we take every bits and pieces of the different tissue cells and parts of the cell, and we say, okay, this is the form which is expressed here of that particular protein. But it's a project, even if we call it a pilot project. The Chromosome 21 will be continue for a very, very, very long time to be the rooms of chromosome 21, the genes. They will continue for a very, very long time to be furnished in respect to looking at this as a room. So more and more detail will be added, more and more information will be added, where they are expressed, which forms, etc. And also, of course, how this relates to disease.
What's the scale of this project? How does it compare to sequencing a chromosome? Or how does it compare to the Human Genome Project as such, in size and cost?
In size and cost, you have to remember when the Human Genome project was executed, they had a budget of $1 billion or more. Now, today, it costs 50,000 or less, and we are approaching going down to tens of thousands, etc., within a couple of years in having a complete genome. So the size and scale in terms of money for doing something like this is something that if we put a number on it today, let's say for chromosome 21, we would need maybe to do a realistic stab at it, something on the order of maybe $50 to $100 million to do that. But you can already do a portion of this. It depends on the detail, the level of details, etc., that you want to have. So if you say we're going to do chromosome 21, and we're going to do that in a number of tissues. For some countries that have picked chromosome, they have decided to map them in tissue cell lines that are immortalized cell lines. It all depends on the level of detail that you want. But you already have the genome, which is the building. So how much furniture do you want to put into each room? That's basically we're never going to be there, because you have to appreciate that you have, if you take a typical gene will give rise to a typical protein. That protein will have splice isoforms. It will have gene-derived isoforms. It will have all these different variants that comes through these genetic variations of the genes that I was referring to, which we termed SNPs, and here then amino acid relevant SNPs. But on top of that, there's a whole universe, which is the post-translation modifications, and these are not. Some of them are binary in form. That phosphorylation, you either have it or you don't, and you can have that on several residues within the protein. But if you take a post-translation modification like glycosylation is enormous complexity. And a protein in a given location can have one type of glycosylation structure, but the same protein in a different location can have a completely different oligosaccharide structure, glycosylation structure. And this also are structures that will change in relation to disease. So pick your number. It's okay,
100 billion. It's going to at least 10 billion, right? This is
that's where I think we need this pilot project to see how much it costs for how much benefit, and how much detail can we generate for how much cost. In fact, so there is an estimate out there that certainly using mass spectrometry methods and some antibodies to cross-check what the mass spectrometry is doing, that a preliminary estimate would be around $15 million now to get a first-pass coverage of the proteins of chromosome 21 in their major sites of expression, in the major cell types of expression, with their protein partners and with their glycosylation and phosphorylation modifications, the major ones mapped. And so that's a pretty good start. Now, at the same time, because of the disease relevance of chromosome 21, one can test whether or not this information, as it's being developed, is impacting what we don't understand about trisomy of chromosome 21 and the diseases behind the trisomy of chromosome 21 in general. So now I'm going to give you two more examples. So for patients with Down syndrome, they actually have 4 times the rate of type 1 diabetes. They have 10 times the rate of thyroid disease to those in the general population, all because of immune aspects, mainly of what's happening in the thymus. And so the mechanistic insight that we're going to be able to get by saying, okay, the universe is only these proteins, 10% of the genome, the payoff may be considerable insight into not just the leukemias, not just the developmental defects associated with the cardiovascular problems, but also diabetes and thyroid disease and even celiac disease, all of which are linked to this 10% of the genome being expressed at 50% higher level. So there are immediate ways of testing cost for benefit, whether the benefit is biological insight, whether the benefit is identifying the proteins themselves and their major sites of expression, and whether the benefit is clinical relevance. So all of these three happen to be plugged in to chromosome 21. So it really is in my mind, a prescient choice as to what might be the pilot project. And that, in fact, was put together in the original white paper that Tommy, Matthias Hewlin, and I put together a couple of years ago now in order to try to see how this project might be launched.
Could you perhaps be, we're going to have Rudy Aebersold next week at what time? We're doing it early. We're going to go on at 10 a.m. Pacific, 1 p.m. Eastern time. And he'll go on into great detail about the methodology. But perhaps you could describe, we're going after chromosome 21 as a pilot project. How do you go from genes to proteins? How does the mass spec work? And why would you select, how do you select a chromosome-based approach to proteomics? So how does it quickly, like kind of
So as Tommy already indicated, we have, because antibodies are raised and they're being screened to various samples of organs from humans that are available to pathologists right now, where we already have a rough idea of what organs are expressing some of the major known proteins of chromosome 21. We already have transcriptional data which indicate what are the major organs that are expressing the major genes from chromosome 21. And we have insight into the organs most affected by chromosome 21, trisomy of chromosome 21, from the clinicians. So we already have this enormous wealth of data that allows us to focus on what parts of the human body should we be sampling in order to now characterize the proteins and identify what it is that they are, how abundant they are, what their partners are, and what their modifications are. So that information is ready right now. What Tommy and I do specifically to overcome the dynamic barrier, the dynamic range barrier in mass spectrometry, and Dr. Aebersold next week, Rudy, will have techniques that he's developed to overcome that directly at the mass spectrometer. What we do as biologists is we go into the cell and we will pluck out those parts of the cell where the proteins are present at their highest abundance. I'm going to give you an extreme example. If you want to detect insulin in the plasma, it is present at 10 to the minus 11th molar. To give you some idea of how low a concentration that is, imagine that you had one molecule or one protein in the volume of a bacterium such as E. coli. The lowest concentration it could be in E. coli is 10 to the minus 8th molar. Yet insulin in your plasma is present at 1/1000th that concentration. Now, wouldn't you rather characterize insulin if you wanted to detect it in its major site of expression in the beta cell, where in the granule of the beta cell, the secretory granule, it's present at crystalline concentrations, right? Hundreds of milligrams per mil. And so consequently, what Tommy and I do is we don't go and characterize insulin in the plasma. That's an incredibly difficult problem. We go and pluck out the granule from the beta cell where it's present at crystalline concentrations and technically determining its abundance, figuring out from the beta cell what its role is a far easier strategy. And that's how we do it. So in each of these organs, getting the organelles which are expressing the proteins is what Tommy and I do. And then these organelles, the small little organs in the organelles, are then following the methodologies which the entire field of proteomics has built up over the last 10 years to reproducibly and quantitatively characterize the abundances of these proteins in these samples. The way it does that right now is the proteins are broken down into peptides by using a digestion protocol, trypsin. And then those peptides, the sequences of those peptides are deduced by the mass spectrometers, basically by fragmenting the peptides and matching the masses of the fragmented peptides to those of the parent peptide, which was not fragmented by the mass spectrometer. And it just so happens that the mass spectrometer fragments peptides where there are peptide bonds between amino acids. So you end up reading the sequence of the peptide. And so it's a high throughput protein sequencing machine, which just like the new deep DNA sequencing machines, counts the number of times it does that, and that's the abundance of your protein. There are other methods which Dr. Aebersold has refined in order to give you another estimate of the abundance of the protein. And that's based on a mass spectrometer's way of doing an ELISA or a radioimmunoassay. And he'll explain that next week. These methods are robust. They have been well followed over the last 10 years or so, and they're what we use to give us the protein abundances and to deduce the entire protein sequences of what we're seeing. And that's that sort of strategy that's also used to look at the protein partners and also to look at the post-translational modifications. Even the splice isoforms are picked up this way because the power of the new generation of mass spectrometers, just like the deep DNA sequencers, is that they can do so many runs and so many analyses at a tenfold greater depth than they previously were able to do. And now you pick up the splice isoforms where the peptides which match the protein sequences which span two different exons are found, and there you absolutely nail down the protein isoform that you're picking up in your sample. And if there's more than one protein isoform, the mass spectrometer will sort it out and give you the relative abundances of all the protein isoforms. So this is an amazing technology which is available us to do right now, and that is roughly how the technology works. Now, Amos Barrock is a specialist in handling the data of how you now match these to the protein sequences. And that is a whole new part that the bioinformatics community, along with the mass spec community, have developed in tandem in order to make the actual data resource that you get of immediate value to the general biologist or the clinician. So I think that roughly summarizes the technical aspects, although I can go in greater detail, Mark.
That's amazing. It seems like this is the tricorder, right? It's the ability to monitor the presence, the concentration of the units that make up the human body down to exquisite level of detail, and really give you a picture of what's going on. How precisely can it detect a protein? How does it, in terms of mass, so you have a protein of a certain mass, it flies, you chop it up, you fly it through the machine. How does it fly? How far does it fly? How does it stick? How does it detect?
So the late John Fenn, who passed away recently, and other investigators in the field, developed this method of allowing not just peptides, whole proteins, to be able to be sprayed into the mass spectrometer by this electrospray method he and Matthias Mann really pursued in great depth, and allow us then to measure the time that this peptide takes in order to reach basically a detector in the mass spectrometer, and that gives you an estimate of its mass to charge ratio. And so these are methods that are as well established now as a spectrophotometer uses to measure absorbances and deduce concentrations based on whatever is absorbing the material, either for nucleotides or for amino acids or for proteins or whatever. So these methods are now so well established, and the mass accuracy of the mass spectrometers are so great that their mass accuracy right now is routinely 1/10 of a proton in terms of mass that they can pick up. And so this is a technology that was developed by folks like Rudy Aebersold, Matthias Mann, John Yates, John Fenn, which is now just routine for any biologist to use. It really is, in terms of its mass accuracy, able to detect unambiguously peptides of specific sequences and be crystal clear in terms of what it is that it's looking at. So, I mean, Rudy will take you through the workings of the mass spectrometer far better than what I can do, or even Tommy, because at the end of the day, Tommy and I are biologists and use the mass spectrometer for trying to make the biological discoveries that we're supposed to be paid to make.
Just one thing to add to that, John.
Yes.
So one thing that I think one should bear in mind is that there is a continuous, really amazing development. So doing mass spectrometry 10 years ago or 5 years ago, even a couple of years ago, as compared to today, is really like night and day, and this continues. So 3, 4, 5 years from now, what we can do today, we can do even better and even better and even better in the future. And this technology is driven by people who are extremely innovative in the academic arena. But at the same time, one shouldn't forget that companies, the industry is a major force in developing these technologies. And I used to coordinate the European network in microscopy, and there we did this together with the companies because the companies are so embedded in the needs of the biologist in terms of imaging. But that's the same here in terms of mass spectrometry. The industry is really closely embedded with the academic researchers, pushing the technology continuously through innovation. But it's not just academics. There are a lot of smart people within the engineering, within the physics, etc., that all contribute to this enormous revolution.
John, would you? I think we have a few more minutes. It'd be really great to get a little bit of the framework for the Chromosome 21 project. So we sort of now have a grasp of sort of how it works. Is there? And we kind of you brilliantly described how those the diseases, the information that could relate to modern medicine. So I'm wondering, do you have any thoughts or ways to present? Because this is really complex material, by the way. I'm struggling because even with a PhD from McGill, great school, you guys are both from McGill. Worked quite a bit with John too, and he was a great help towards my PhD. And I thank you for this is hardcore material. This is really it takes like a comprehensive understanding of biology to see where and how this gets into the context of medicine, right? So when is the pilot project going to start? Has it started?
So it has not. Well, I mean, I think as Tommy pointed out, antibodies are being made by Matthias Uhlin right now to every predicted protein coding gene. And so those are in progress, and probably the entire genome will be finished certainly within 5 or 10 years by that approach. And where you'll have some sort of antibodies to representative proteins. With respect to launching the project on chromosome 21, or any chromosome for that matter, the Human Proteome organization in Sydney, at last year's meeting in Sydney, launched an effort to say, let's start the Human Proteome project. Next Tuesday in Toronto, there's a meeting of the Canadian Proteomics Network. It's the second meeting. The first one was in Barbados in January that Tommy and I coordinated with them, where they will decide whether or not chromosome 21 is something that Canada should go forward with, or perhaps another chromosome, or perhaps 2 chromosomes. The original white paper that Tommy and I and Matthias Uhlin and other folks put together had proposed chromosome 21 as the pilot project, so that's still my favorite pilot project. And so far, even though the community and the Human Proteome Organization and the Canadian Proteomics Network has said we're eager to do it, the actual execution of the project has not yet begun, other than the fact that in each and every lab, we're getting set up to try to get the samples ready, to try to do the analyses, but to do it in the coordinated way that Tommy outlined at the beginning, where many labs are coordinated together to focus on the samples that are generated simply to really map comprehensively and quantitatively the proteins of chromosome 21, that has yet to be put together, and that's what we're hoping we'll be able to do in a very short period of time. But certainly the meeting in Toronto on Tuesday, next Tuesday, is meant to address that issue with all the funding agencies.
It's amazing. This is one of the, I think, you mentioned a tumor suppressor that there's obviously a tumor suppressor, but it's not clearly understood which it is or how it's working. And if you could pull that out and, turn cancer into a chronic disease rather than a lethal situation, it'd be fantastic. This
and
is
I think
pretty
it's
amazing.
reasonable. I think it is reasonable to hold us to that bar, to say that, well, if you're going to map these proteins, we do demand some mechanistic insight and some clinical relevance. And I think that's reasonable.
Well, if I feel like just throwing money through the camera here. You present a very compelling case for the next stage of what I consider the next stage.
We sell used cars on the side.
Yes, but you understand how those cars work to the atomic level, which means
yes.
It's amazing. It's really great stuff. You know, this project that you guys are doing. When I first read the paper in Nature Methods, it really is exciting that you guys are taking on an ambitious venture and doing it with aggressively and presenting it with a tremendous level of compellingness that it's moving biotech forward in a significant way. I think this is kind of the perfect show, although the material is fairly rich in information here. They require a second listen. I'll probably listen back and get a lot out of it. It defines very well what Futures in Biotech is about, and trying to understand is where medicine is going and where our understanding of human biology is going. And you guys are really at that frontier. So I really thank you for coming on the show and sharing your time with us.
Well, thank you very much, Mark. We're really grateful for this opportunity.
Yeah, thank you, Mark.
I think we've got a great show. That was some very insightful thoughts and very. It's almost like the first part, And again, we're going to record with Rudy Aebersold next week. But even then, I think this really is going to be a reoccurring theme over the next year. And I really wish you guys the best at really moving this forward. It's going to be really great. I can't wait to have a mass spec in my lab. And John, I'm going to be calling you.
Okay.
So I will let you go. Before we go, I'm going to just go through the thank yous. So we do a recording so we can edit here. Burke, if you could put a marker. I'd like to thank our guests, Dr. John Bergeron. He's professor and chair of the Department of Anatomy and Cell Biology at McGill University. Thank you, John. I'd also like to thank Dr. Tommy Nilsson. He's professor in the Department of Medicine at McGill University and Canada Research Chair in Proteomics and Systems Medicine. Thank you guys very much for coming on the show. This was a lot of fun.
Thank you for having us.
Thank you, Mark.
I'd also like to thank Brendan Glenn, who's from Med City News, who's doing a piece on Futures in Biotech and on me. There's Brandon. Go check out Med City News. There's a really great source for what's going on in medicine, certainly in the Midwest as well. I'd like to thank Burke McQuinn for handling the audio and video boards and recordings today. Thank you, Burke. And the team that make this possible: Leo Laporte, Lisa Kensal, Frédéric Louis, Eileen Rivetta, Tony Wang, Mike Taylor, John Slanina, Jess Stewart, and Jason Howell, and the rest of the team in Petaluma. I haven't met everybody there yet. Lastly, I'd like to thank Phil Pelletier and Will Hall for the opening and closing themes. If you have any comments or suggestions or questions, you can reach me at marc@twit.tv. It's MARC@twit.tv, or on Twitter at Marc Pelletier, MARCPLLETIER. For Futures in Biotech, I'm Marc Pelletier.